Good morning, students. Myself, Major Komal Katyal, Department of Zoology, GK Gowani Government College, Bhinmar. Today, we will discuss about the antigen and antibody reactions. Now, what are the antigen-antibody interactions and how we can characterize all these interactions? Let us discuss here. Now, they are irreversible or reversible. It means they are reversible but specifically non-covalent biochemical reactions. The bonding is the hydrogen bonding, and as you know, hydrogen bond is a chemical bond in which a hydrogen atom of one molecule is attracted to an electronegative atom of the another molecule. The electrostatic bonds is a valency bond in which two atoms attracted by electrostatic forces transfer one or more electrons between atoms. The van der Waals forces are the forces which act between non-bonded atoms and the molecules. The hydrophobic bonds, the attractive force between molecules due to close positioning of the non-hydrophilic portion of the two molecules. Now, these antigen-antibody interaction can be represented by the following formula. Ag here depicts the agglutinogens, that is the antigens. Ab is the antibody or agglutinins, and as I told you, they are the specifically reversible reaction where there is formation of a complex between antigen and antibody, and the K1 and K2 are the constant of association is the K1 and the K2 is the constant of dissociation. It means during association the constant is K1 and during the reversible reaction the K2, which is the constant of dissociation. Now the affinity, अब हम बात करेंगे बंधुता के बारे में. What is the affinity? It is the strength of the reaction between a single antigenic determinant, that is the epitope, and a single combining site on the antibody. It means reaction between the epitope and the paratope, or it is the association constant of binding, which can be depicted as Ka. So Ka is the K1 upon K2. Depending upon the affinity, the high affinity and the low affinity, we can determine this association constant. The valency, as I told you earlier on in my first lecture, is the number of epitopes present, while the avidity is the collective affinity of multiple binding sites. It means affinity plus valency. We have discussed about it in my earlier lectures. Now, before beginning the types of antigen and antibody reaction, there are certain features which are very common in the antigen antibody reaction, like the formation of the immune complex, which I have already told you. The second one is the specificity, which we have already discussed. Then the binding sites, which I have already told you, like the epitope and the paratope. The binding forces, which we have just discussed, the various type of bonds. They are the intermolecular forces governing it. We have already discussed about the affinity and the avidity. So all these are the main feature. Then certain bonus effects are also present during the antigen antibody reaction, and these bonus effect actually binds with the antigen, but also they bridge the antigen with the antibody. It means they actually help in the antigen antibody reaction. It means it is a phenomenon of giving extra strength to the antigen antibody complex by binding of two antibodies to the two antigen molecules. It is the bonus effect. Then certain cross reactions are there, about which we have already discussed in my previous lecture. Now let us discuss about the type of antigen antibody reaction. The precipitation, the second one is the agglutination, third one is the neutralization, fourth one is the opsonization, then antibody dependent cell mediated cytotoxicity, and the complement activation membrane attack complex. Now the consequences of antibody binding, as you can see, the two antigens are shown here, one the virus and other one is a bacterium. Now during neutralization, as you can see, it blocks the viral binding sites or coats the bacteria in the case of opsonization. When we talk about the agglutination, it means the antigen bearing particles such as microbes, and these antibodies bind with the antigen. Here they are the bacteria. The precipitation of soluble antigens, it means certain toxic substances which are released by the microbes or the pathogens invading our body, and these soluble antigens are precipitated by antigen antibody reaction. The complement fixation, it is the activation of a complement complex where a foreign cell leads to the cell lysis. Now neutralization, opsonization, agglutination and precipitation ultimately enhances the process of phagocytosis. It means the cell mediated immunity. The macrophages, which are the main phagocytes present in our body tissue, are responsible for carrying out the phagocytosis. Let's discuss about each reaction. First, the precipitation. It is the reaction of soluble antigen with the soluble antibody. The reaction result in the formation of antigen antibody complexes, as you can see here. There is formation of complexes. Now the quantitative precipitation reaction, the varying amount of antigen are mixed and incubated with constant volume of the antisera. Now the precipitate is measured, and the amount of precipitate depends upon first the ratio between antigen and antibody, the avidity of the antibody, and the three zones we can see. First, the zone of antigen axis, then the insufficient antibody, too small complexes to precipitate. The second, the equivalent zone, the large lattice is formed and the visible precipitate are there. And then zone of antibody accesses, not enough antigens are available, too small complexes to precipitate. All which I have discussed in the previous slide are depicted through graph here. You can see the region of antigen axis, antibody axis and the equivalent zone also. The agglutination, the antibodies can bind the cross-linked cells or the particles, and there is aggregate formation, and these aggregate actually entrap the microbial invaders. The most suitable antibodies are the IgM and IgA for this purpose. This is the case of agglutination. You can see how the process is going on. Now, the application of agglutination, the most common which we have observed in the case of qualitative agglutination test or heme agglutination, that is the determination of the blood types or antibodies of the blood groups. The quantitative agglutination test includes the titration. The passive heme agglutination, the erythrocytes are coated with a soluble antigen, that is the viral antigen or a polysaccharide or a hapten, and it is used to the coated RBC in an agglutination test. Here you can see the example. The Coombs test is also one. Now the neutralization, it is the binding of antigen to the microbial epitopes or the soluble molecules. That is the toxin which inhibits their binding to the host cells. The most commonly used antibodies are the IgG and IgA, and they are used to identify the toxins and viruses. Then the opsonization. It is a process by which a pathogen is marked. It means the tagging process for ingestion and destruction by the phagocyte. It means first it is marked so the phagocytes can easily attack. The antibody dependent cell mediated cytotoxicity, it is the coating of an organism can attract the phagocytic cells like the natural killer cells, eosinophils, and other cytolytic cells. The organism can be bacteria, protozoan, or parasitic worms, and the cells use the cytolytic mechanism to kill those organisms. Now this is the process of destruction of large parasite. So we have discussed about the precipitation, agglutination, the opsonization, and neutralization. Now certain reactions which are known as the flocculation. Now flocculation is basically an antigen antibody reaction which is brought about by the exotoxins and the endotoxins, and these reaction produces floccules which do not sediment but remain dispersed in the sediment. The flocculation is somewhat like precipitation, but the precipitate will not sediment, and here the reaction of which produces a floccule but they remain dispersed in the medium. Apart from it, there are certain immunofluorescence reactions are there where direct and indirect methods are used. The direct method is actually used for the detection of rabies, while the indirect method is used for the tissue antigen and for the specific antibodies. So these are the basic features of antigen antibody reactions, and the antigen antibody reactions are later on used to determine the type of antigen or type of pathogen infecting our immune system. So this completes my lecture on antigen antibody reaction. Thank you.